Assalamu alaikum. My name is Rida Masood and I'm a student of Mphil first semester from the Department of Microbiology, Kohart University of Science and Technology. The topic of my seminar is bioremediation of oil contaminated soil. So this is the front page of that research article from which I have made my presentation. Now let's come toward the outlines, which includes objectives, introduction, material and method, results and analysis, discussion, conclusion, and references. Now the objective is to study the microbial remediation of oil contaminated soil. In this we are going to study that how microbe has played their role in order to degrade oil and make the soil decontaminated. Different kind of media were used, different kind of method were used, which I'm going to discuss them in the upcoming slides. Now let's come to the introduction, which says that petroleum carbons are a major energy source as they cause leakage and disturb the natural ecosystem because they produce different kind of toxic compound which are harmful to humans and the environment. But we are fortunate that their degradation is possible, otherwise life on Earth would be impossible. So the present study focused on the ability of two strains, that is G40 and G94, which were used in order to degrade oil. And these two strains were obtained from the fungal species, which are named as Bacillus lactosporus and the Candida tropicalis. Now the materials includes experimental samples which were collected from the Agriculture College of Yonsei University. Air was used to dye the samples, and the media that has been used in the process are beef peptone liquid medium and the potato sucrose liquid medium. Because both strains were obtained from fungal species, that's why these media were used. Now, the strains which were collected and isolated were basically taken out from Kongjang Ganboha oil field. These strains were then treated with beef peptone liquid medium and a potato sucrose liquid medium on which they were activated. If we talk about petroleum contaminated oil, then oil was first dissolved in petroleum ether and then soil were added into the soil. It was kept inside it for about 7 days, on which it gets dries up, and after that it was observed that petroleum ether was completely volatilized. Now different method has been used to check the availability of alkaline hydrolyzable nitrogen for the available phosphorus for the oil removal rate. So for the alkali hydrolyzable nitrogen, alkaline solution diffusion method was used in the process. For the availability of phosphorus, sodium bicarbonate extraction method was used, and for the oil removal rate, gravimetric method was used. Now the experiment basically depends on the four treatment. In the first treatment, G40 strain was taken out and was inoculated, and the amount of inoculation for G40 strain was 8%. And in the treatment second, G94 strain was inoculated, and its inoculation amount was 4%. And in the third treatment, both strain were added together, and in the control, no strain were added. The experiment is basically carried out in tissue culture bottles. In each bottle, 20 gram of petroleum contaminated soil was added with the addition of 20% of boron. Distilled water was also added into the soil so that the soil moisture content was reached up to 35%. Then after that, all bottles were autoclaved and the degrading microorganisms were inoculated and placed at 35 degrees centigrade in the incubator. Now, different soil samples were used to determine and study the soil alkali hydrolyzable nitrogen, available phosphorus, and oil content. These soil samples were studied on different days like first 5, 10, 15, 20, 30, 40, and 100 days. Now, the result and analysis for the alkali hydrolyzable nitrogen is shown in this figure, and the graph is basically between time and alkaline hydrolyzable nitrogen content. The different treatment groups are indicated by different colors. Like control is shown for shown in blue color, red color is shown for G40 strain, green is shown for G94, and purple color is shown for G40 plus G94 strain. With the increase of time, the content of alkali hydrolyzable nitrogen in the three treatment group are increased from first to 10 day. After that, no change was observed until 100 day. G40 plus G90 treatment group was much higher than that of G40 and G94. So it was observed that G94 and G40 strains, they were work together very efficiently than the individual strains. Now for the available phosphorus, the content is shown in this figure. Basically, it did not change from first to 40 day. After that, a sharp change was observed. Again, it was observed that G40 plus G94 treatment was much higher than that of G40 plus G94. Similarly, for the oil removal rate, up to 15 day, the rate was increased very slowly. Onward 15 day, further increase was observed. So it was uh, observed that G40 plus G94 strain were again much higher than the G40 and G94 strain. And their amount is given for G40 and G94 and G40, the amount is 29.08%, and 31.09%, and for G40 plus 94 strain, the amount was 32.68% on 8 day. Now the discussion. The process of oil removal rate is very complex because of the presence of microorganisms. So its rates not only depend on the microbial community, but also depends on its composition and the number and the availability of total petroleum hydrocarbon. Besides this, it's also depend on the structure of the soil, parent material, and the moisture. So in this experiment, it was observed that microbial degradation of petroleum efficiency was low. So in order to achieve higher degradation efficiency, different amount of nutrients could be added with proper amount of water, and some different kind of microbes can also be used for higher degradation efficiency. So at the end conclusion, what we have concluded from this experiment, that the oil removal rate of G40 plus G94 was much higher as compared to G40 and G94 strain. So this is what it was concluded. At the end, I have added the references, and this was all about my presentation. Thank you so much for listening and watching to my presentation.